Hi, my name's John Higgins, and this is a seminar on the HAB trap and removal process. It's a National Science Foundation funded technology. It's for removal of harmful algae and cyanobacteria cells and blooms from water bodies. In the fall of 2020, we received a National Science Foundation grant for our research for the HAB trap and removal process, and which is wonderful. And NSF, I just wanted to promote NSF a bit here as it's so important to us for this process. And their mission is to promote the progress of science. And I can attest that the review process that they have for proposal applications is very rigorous and time consuming, and you really have to have your soul and your heart into it in order to complete that process. And so the NSF, they are designed to, foster and promote the best ideas for the best science, for the best returns for our nation. And we're just so deeply honored to have received recognition and funding by NSF for our work in research and development. And we also just happen to have also received honors on Inventors Day in 2019 by the Boston Bar Association in Massachusetts for the same patent. And as a group, with people from MIT and Draper Labs, we also received recognition from Governor Baker. So I do recommend that you check out NSF's merit review process, and I have a link here to that. It's really a wonderful video and informative process, and it's also available on our sister website, www.peapodtech.com. This is just an illustration because it helps of the HAB trap and removal process. This is an open water technology. It's made out of fabric mostly, and here you have a floating edge which goes around. These are what I call the trap area. And this is like the collection area, and this is the influent for the apod. And you can put a screen there to keep non-target biota out, like turtles and frogs or fish. Here's a picture of an early prototype, emphasis on early, and, but it just helps to illustrate different components of the apod because there's some visual, you can see visually collecting the cyanobacteria. So this is the apod trap area. This is the collection member. Here's some of the scum coming in. It isn't just the surface, it's also for cyanobacteria, the suspended cyanobacteria that you don't see on the surface. But I just like this photo because it is visual. And on the background here you can see an active collection member, and this can be drawn back to force in a targeted area of the water body that has the HABs of interest and to force those to be trapped into the apod. This is an example of kind of a larger scale use for the apod, and this is it's a 300-acre water body. And the areas in magenta are areas of concentrated HAB cell densities. And those are the type of areas that once you identify them, or you can anticipate them sometimes, you can position and use the apod to target those directly. And here's an example of kind of how that would be set up. So here we have an inlet stream, some natural water flow, and there's an area where those HABs tend to collect. So I would position an apod unit there in passive mode to just collect those HABs into the trap area passively. And over here I have an apod set up in more of an active mode. This may be an area like wind-blown HAB deposition accumulation, and the active member of the apod is extended outward around that entire targeted area and slowly drawn back to force the targeted HABs into the trap where they can be removed. This is what the HABs look like when they're removed. This is kind of before. This is actually a cyanobacteria. And this is after. It's like an algae. It's like a shaving cream. It's just really concentrated. It's great. And that can be removed with the excess nutrients and the toxins from the water body very quickly. And as we'll talk about later, the removal process does not release HAB toxins, which is great. So our field trial location number 1, these are results through 2020. We actually started this before the National Science Foundation grant. This is our location number 1. And it's a park pond. It's 4 feet deep. It's only 0.4 acres, which is great because it allows us to have a better control on the metrics of evaluation, because there's just less variability for a large, like you would get in a larger water body. This location had a history of cyano-HABs, scums, and benthic algae mats floating up, and they would treat it twice a year, and you really couldn't tell by looking at it. They used flocculants, aluminum sulfate, algicide, copper sulfate, and they used herbicides for some of the wetland plants around the edges. This is a landscaped park pond. And as part of in the course of 1 year for our prototype, we captured and removed 320 dry weight. We dried out the cyano-HABs from the pond, and as well as their nutrient content and toxins. And that had a resulting decrease in total phosphorus content of sediment by 85%. I'm going to have a summary table that follows this slide that shows you those lab results. Kind of corresponds to a change in pond status from eutrophic to oligotrophic. And interestingly, we also had a reduction in the total phosphorus content of the algae recovered itself over time, from luxury uptake to growth limited, and there's a decrease in 98%, which is great. So here's our summary table for our baseline and year 2019 results. It's just prior to the National Science Foundation work, but it is in support of that. And we have a summary later on of some of the work we've done in 2020. But what we have here is our sediment up on top. And in 2017, we did a baseline sample, sediment. And for sediment samples, what we did was we took 3 transects, divided the pond into thirds of the central part, and we took discrete samples on each transect and composited them together. And so our baseline total in top 6 inches, baseline total phosphorus concentration was 1,420 milligrams per kilogram. And then as you see here, as you go on through time, we did an initial, this is down here is our cyanobacteria samples. And you can see we had, this is our baseline just a year after we took our baseline sediment sample. We had 12,400 milligrams per kilogram of total phosphorus in the algae. And that's that luxury uptake, really high concentrations. And in that following spring, we took another baseline sample, and we had removed some HABs in 2018 in the fall, and we had a corresponding decrease of about 50% in sediment just from that one removal period. But we still had elevated phosphorus in this cyanobacteria that we recovered in the beginning of 2019. So those cyanobacteria do seem to be picking up the nutrients from the sediment surface and holding on to them. And so in 2019, we did a really large, basically a summer-long APOD removal process of HABs, and we had a corresponding decrease in sediment down to 219 milligrams per kilogram, and that is the 85% decrease that I mentioned before. And we saw a similar decrease, pretty notably, for the cyanobacteria themselves. This is the 98% decrease. So at the end of our larger APOD trial, 283 milligrams per kilogram. And so in summary, part of 2020 results, National Science Foundation supported. We were actively measuring and monitoring the cyanobacteria cell counts to see the timing for the use of the APOD, and the cell counts increased and plateaued just at that first week of September, and we deployed the APOD at that time. And within 3 days, we closed off APOD. There was a storm coming. We wanted to see what we had for data. So we deployed it for 3 days, passive use, favorable conditions, and the APOD had concentrated the microcystin toxins by tenfold in the trap area as compared to the rest of the pond. And we closed off the trap and we initiated removal actions. And one of the National Science Foundation feedback we got was to kind of test to make sure we weren't releasing toxins as part of the removal process, so we tested for that. We had both a midpoint and 2 effluent samples within the trap area itself, and we had no, even by the midpoint, the first treatment step, we had no detectable microcystin contents, and the HAB biomass concentration was decreased significantly. It was visually apparent within 10 minutes, and we had no cyanotoxins in our either fall and effluent samples. And we took those recovered HABs and we did a compost trial with air drying and photodegradation of that material, that kind of the algae shaving cream I showed you before. And we had a decrease in microcystin toxin content by 50% in 1 week. And the control sample had no change, so that favorably showed that one, we were able to remove HABs and their toxins from the water body. We concentrated it, removed it without releasing toxins, and as part of the reuse situation we evaluated, we had a decrease in those same cyanotoxins within 1 week. We'll continue that testing in the fall in this. So one of the benefits of doing innovative field trials is you do gain some insights into different aspects of our environment relative to nutrient enrichment and harmful algae blooms. And so some of the things that we took away from this trial here so far was that for this particular location, despite years of biannual aluminum sulfate treatment, we still had concentrations in phosphorus that were rapidly decreased during our trial. They were uptake by the HABs themselves, which were removed. And so that pretty well documented that the aluminum sulfate treatments really didn't effectively bind up those nutrients in sediment. So the other thing we have from our testing was clear that HABs gather, build up, and store ore grade concentrations of nutrients. And for HABs, that's iron, nitrogen, phosphorus, and sulfur particularly. And in that regards, they are like, we consider them like eco-miners that in turn can be targeted and removed with the APOD technology. And as such, the APOD technology really is an ecologically sensitive way to remove the HABs, their toxins, and the excess nutrients from water bodies. But we're essentially working in tandem with the HABs. They remove all the toxins, all the excess nutrients, and we remove the HABs. So our testing also showed that we had elevated concentrations of sulfur in the HABs. And we did check on that, and sulfur is an essential macronutrient for HABs, and it's nearly equivalent by ratio to phosphorus. And there are some other interesting interactions that sulfur has with biogeochemical cycling of other nutrients. And please go to our website, our sister website, www.ppodtech.com, to look at our lake iron nodule presentation for more information. 2021. So we're going to be continuing in 2021 with the APOD HAB trap removal process and National Science Foundation supported work. And some of the additional metrics that we're going to be looking at are different water body conditions, scales, types of water bodies, rivers, lakes, ponds, estuaries, possibly, hopefully. Other metrics are different types of HABs, the picos, the bloom-forming colonies, some of the deeper occurring cyanobacteria types, different biogeochemical conditions of water bodies, and also areas of water bodies that have a history of different treatment efforts. We can kind of come in and see how we can improve things. So we are looking for additional field locations as part of this National Science Foundation program. So So please do contact us directly, and there's some information at the end of this presentation for contact, and appreciate it. And so just in summary of the HAB technology, the HAB, it targets, captures and permanently removes HABs, their toxins, and excess nutrients from water bodies. Now, it doesn't just suck out the entire water body. You can actually use it to specifically identify areas to be removed, target those, and you can include measures, deterrents that keep non-target biota from being trapped. Even if they are trapped too, you can inspect the HAB trap to make sure it's predominantly what you're trying to remove. And if it isn't, if it's mostly good algae, you could just release that back into the water body. We are going to be further automating the HAB process, particularly for areas of HABs that are more toxic than others, just to limit end user time and also to facilitate collection and disposal. And the HAB, so what's really neat about this technology, unlike other technology or approaches out there, this is fully scalable. It's readily deployable. You can use it for rapid control, removal, and prevention of risks due to HABs. It is cost-effective, sustainable, and eco-friendly. So this is a patented technology. It is specifically designed by me as part of the patent to be eco-sensitive. It's an open water technology. It doesn't just dredge sediment. It doesn't, damage, buffer zone vegetation. You can limit its bycatch, and the user can inspect the contents of the trap area to decide whether that's to be removed or not, and if you don't want to remove it, you can just release the contents back into the water body. This is my contact information. There is additional information on peapodtech.com, some downloadable reports. There's some previous webinar slides that I've done in the past as well. And this is kind of how it all started for me. This is a picture of me with this thing called the lake iron nodule. Please give me a call if you have any interest in learning more about those. Thank you very much. And again, if you have locations with HABs, please give us a call. We can discuss whether they'll be suitable or not as part of this National Science Foundation trial process. Thank you very much.